Imagine looking at like a sprawling, fully illuminated map of a bustling city.
Oh, wow. Okay, I'm picturing it.
Right, but imagine that instead of just seeing the buildings and the streets, you can instantly see exactly what every single person inside those buildings is reading or saying or doing all at the exact same time.
That would be, an overwhelming amount of information.
It really would. But tell you what, that is essentially what scientists have just achieved. Not with the city, but with the mammalian brain.
Which is just a staggering leap forward.
Yeah, it really is. And so the mission of our deep dive today is to unpack this totally groundbreaking 2026 paper from the journal Cell.
Right, the one introducing RT&T-AMP-MERFISH.
Exactly. It's a bit of a mouthful, but we're going to break it all down.
Yeah.
Because this team literally mapped over 33,000 RNAs, including 23,000 genes and 10,000 highly specific isoforms, completely intact inside single cells.
And to really grasp why that's a big deal, you have to understand the historical bottleneck we've been stuck behind, for decades.
Right, because traditional sequencing gives you this incredible genetic picture, but it completely shreds the spatial context. It's like throwing a masterpiece puzzle into a blender. You have all the pieces, but you have no idea how they fit together.
Exactly. You lose the neighborhood. And traditional imaging shows you exactly where things are, but you can only look at a few genes at once. So you have the neighborhood, but you're blind to the actual conversations happening inside it.
And solving this matters so much, doesn't it?
It changes everything. Because as this paper proves, a cell's physical neighborhood entirely dictates its function.
That is huge. So to understand this map they drew, we first have to understand the completely new camera they used to take the picture.
Right. The baseline here is MERFISH, which is multiplexed error-robust fluorescence in situ hybridization.
Such a long name.
It is, yeah. But basically, instead of one color per gene, you use fluorescent barcodes. But unamplified MERFISH has this really severe physical limitation.
Like a real estate problem.
Precisely. To get a signal bright enough for the microscope to actually see it, you have to physically bind about 30 different fluorescent probes along the length of a single RNA molecule.
Wow, 30.
Yeah, 30. Which means if the transcript is too short, you just literally don't have the space to stick 30 probes on it.
So it just stays invisible.
Exactly. Which is why they introduced this new RT&T-AMP chemistry. The AMP stands for amplification.
Okay, let's unpack this, because this is where the molecular engineering just gets wild. They start by fixing the tissue, right?
Right.
But they use glyoxal instead of standard formaldehyde.
Yeah, glyoxal is crucial because it preserves the RNA integrity and the physical architecture far better. It basically freezes the city.
So every transcript is locked right where it was.
Right. Then they send in a poly-T primer. Since mRNA naturally ends in a poly-A tail, that primer just perfectly docks onto the immobilized transcripts.
Setting the stage for the reverse transcriptase enzyme.
You got it. The enzyme uses the locked mRNA as a template to build a complementary DNA strand, a cDNA, right on top of it.
But reverse transcriptases have this quirky property, right?
They do. When they finish, they don't just fall off cleanly. They actually add a tiny, non-templated overhang of 3 cytosine nucleotides.
A little CCC tail.
Exactly, just hanging off the end.
Yeah.
Usually that's a nuisance, but here they weaponize it by introducing a template-switching oligo, or TSO.
And the TSO has a GGG sequence that locks onto that CCC tail, like a molecular handshake.
Yes, and attached to the other end of that TSO is a T7 promoter sequence. So the enzyme switches tracks, reads across the TSO, and basically welds a start button onto every single transcript.
The T7 promoter.
Right. Finally, they add T7 RNA polymerase, which is this incredibly fast enzyme that hunts down those start buttons and turns out dozens or even hundreds of RNA copies right there on the spot.
Like a biological printing press. But wait, I have to push back here.
Okay, sure.
If we use this T7 megaphone to print thousands of copies inside a tiny cell, doesn't it just become a blurry, crowded mess? How do they read it without all the signals overlapping?
It sounds like it would be a mess, right? But that localized crowding is actually the secret sauce. Because you create this dense ball of identical copies anchored to one spot, you don't need 30 probes anymore.
Oh, because the signal is so concentrated.
Exactly. You can get a crystal clear signal using as few as 1 to 6 probes. It makes detecting tiny short sequences possible, and it's vastly cheaper.
That is brilliant. Okay, so let's do a quick recap of this first section for you listening. First, we freeze the cells in place with glyoxal. Second, we use reverse transcriptase and a TSO to install a T7 start button on every transcript. And third, we use that T7 megaphone to print a massive readable cluster of copies right on the spot.
Perfect summary.
So before we move on, I want to throw a question out to you, the listener. Think about your own life. When was the last time you had to isolate and amplify a tiny hidden signal to make sense of a massive problem?
Oh, that's a great question.
It's good to keep in mind as we go.
Well,
But all right, moving on. Now that we have this ultra-powerful biological megaphone, what do we hear when we actually listen to the brain?
Well, they mapped 23,000 genes, and what they heard validates why spatial context is everything. The brain isn't a homogeneous soup.
Right, it's organized.
Very organized.
Yeah.
It's segregated into precise spatial modules. They found massive groups of genes that are strictly co-expressed based on their physical coordinates.
And a really cool example from the paper is the LSUR, right? The lateral septal nucleus rostral part.
Yes, the LSUR. It's a subcortical area deeply tied to emotional reactivity and social behavior.
Right.
And within the LSUR, the data revealed this highly specific module of 207 genes. And when you look closely, you see this localized biochemical tug-of-war happening.
Yeah, this part blew my mind because they mapped the co-expression of a receptor called GLP-1R right alongside a calcium channel called TRPC4.
Both of which are heavy hitters for social interaction circuits.
Exactly. But then living right next door, physically in the exact same neighborhood, is a gene called RASD1. And the battleground here is cyclic AMP, or CAMP-AMP.
Which is a huge stress messenger in the brain.
Right. So GLP-1 activation spikes CMP levels, which drives the circuit forward, like stepping on the gas. But RASD1 does the exact opposite. It plunges the KMP levels down. It's the brakes.
So we are literally visualizing the physical architecture of an emotional rheostat. We're seeing how the LSR calibrates our stress responses in real time.
And honestly, if you're listening to this and you've ever felt the physical exhaustion of chronic stress, this exact tug of war is physically remodeling circuits in your brain right now.
It really is.
Yeah.
And this spatial remodeling goes way beyond neurons. The paper's data on smooth muscle cells, or SMCs, is probably the best proof of this shape-shifting concept.
Yes, the construction workers. I use this image memory trick for SMCs. Usually we think of them as the brawn of the blood vessels, right?
Exactly. Their traditional job is to wrap around vessels and contract to maintain blood pressure. Muscle work.
Right, and the paper showed that when SMCs are densely packed around vessels, they act purely as muscle. They heavily express genes like ACTA2 and MY11 to contract.
But here's the crazy part. When those exact same cells are sparsely distributed, meaning they aren't crowded by neighbors, their entire identity shifts.
They drop the hammers and pick up the blueprints.
Yes, they drastically downregulate those contractile genes, and instead they upregulate a completely different module, like CXCL12 and AGAL7.
And CXCL12 is a chemokine.
Right. It signals endothelial cells to migrate. The cell literally stops being a muscle and starts acting as an architect to build new blood vessels.
Yeah.
Angiogenesis.
So if we connect this to the bigger picture, it's the exact same cell type, same DNA blueprint, but its functional reality completely flips depending on its zip code.
Exactly. The density of its neighborhood changes its entire identity.
Okay, time for another quick recap. 1, the brain organizes gene expression into highly specific spatial modules, like the LSR balancing stress. 2, cell density physically changes gene expression, turning muscle cells into architects. So, question for you listening. How much does your immediate environment or your own zip code change the way you express your own identity?
I love how this biology maps onto human behavior.
It's all connected, but okay, we've mapped the genes, but the researchers went one massive step further into this hidden layer of complexity. Isoforms.
Ah, the secret menu of the genome.
Yes. So let's define this first. Think of a gene as the base model of a car, like a Honda Civic.
Okay.
Standard sequencing tells you, hey, we have a Honda Civic in this cell. But an isoform tells you the specific trim. Is it a sedan? Does it have a spoiler? Is it manual or automatic? They are variations of the exact same gene.
Right, and biologically, these trims are created by alternative splicing. As the cell processes a transcript, it might intentionally skip a section or start at a different spot.
Which slightly changes the final protein.
Exactly. It might change how the protein folds or how long it survives. But historically, spotting isoforms in intact tissue has been nearly impossible.
Because they only differ by tiny, tiny sequences, like a few dozen nucleotides.
Right. And going back to the bottleneck, you can't fit 30 probes onto a tiny splice difference. Yeah. But with RT&T-AMP, because you only need 1 to 6 probes, the researchers could finally see them.
And they mapped 10,000 of them. Let's talk about the choroid plexus, because the specificity here is wild.
So the choroid plexus is basically the brain's water filter. It produces cerebrospinal fluid.
And it turns out it doesn't use the standard genetic base models. It uses its own highly specific trims.
Yes. Genes like SLC4A10, which is a sodium bicarbonate transporter, or EDP1A1, an ion pump. The choroid plexus uses completely unique splice variants of these genes to fine-tune how it filters ions.
We see the same thing in the hippocampus, right? The memory center.
Oh, absolutely. The CA1 subregion uses entirely different isoforms of the same genes compared to the CA2 or CA3 regions sitting right next door.
Okay, wait, let me play devil's advocate here.
Go for it.
If a gene, like an ion pump, is already doing its job, why does the brain bother going through this incredibly complex, error-prone splicing process to manufacture 10 slightly different trims for different rooms of the house? Is this just biological noise?
It's definitely not noise. It is extreme evolutionary specialization. Think about it. A neuron in the CA1 region fires at different frequencies and faces different demands than one in CA3.
So they need different hardware.
Exactly. A slightly different isoform might change the refractory period of that specific neuron by a fraction of a millisecond. It fine-tunes the entire memory circuit.
Wow. Okay, recap time. 1. Isoforms are specific trims or variations of a single gene created by alternative splicing. 2. Regions like the choroid plexus and hippocampus use highly unique isoforms to optimize their local functions. So, question for you. If you were to look at the isoforms of your daily habits, those tiny, almost unnoticeable variations, which ones yield completely different outcomes for your day?
The devil is in the details, right?
Always. Okay. Moving to the final piece of the puzzle. Cells aren't just shape-shifting and changing trims in a vacuum. They are constantly talking to each other.
The cellular switchboard, this is where they mapped about 1,500 ligand receptor pairs across the tissue.
Right. So a cell releases a signal, the ligand, and a neighbor catches it with a receptor. Let's look at the opioid signaling in the striatum.
This is a beautiful example. They mapped the ligand pidin, which makes dynorphin, and its receptor OPORK1.
Usually we hear opioid receptors and think painkillers or addiction.
Right, but naturally in the striatum, this pathway acts as a localized traffic cop. The striatum uses medium spiny neurons to process motor and reward signals split into D1 and D2 pathways.
And the map showed that pidin is heavily expressed by the D1 neurons.
Yes, but the OPORK1 receptors are sitting on both D1 and D2 neurons right next to them. This physical layout confirms a mechanism called lateral inhibition.
So if I'm understanding this, when a D1 neuron fires to take an action, it releases dynorphin. That dynorphin hits the receptors on its neighbors and actively suppresses them. It literally shushes the competing neurons.
It forces the competition to quiet down, improving the signal-to-noise ratio.
That is so cool. But the absolute craziest part of this switchboard data is the Notch1 signaling pathway.
Oh, the Notch1 data is profound. Notch1 is this classic receptor involved in development.
Right. I excitedly wrote in my notes that it's like a smartphone. So every astrocyte in the brain carries the exact same smartphone hardware, the Notch1 receptor.
I like that analogy.
But depending on which city the astrocyte travels to, the phone connects to a completely different local cellular network to get its instructions.
And the data perfectly backs that up. In the cortex, Notch1 receptors on astrocytes talk almost exclusively to a ligand called CCN3, which is broadcast by local excitatory neurons.
That's the local network for the cortex. But down in the striatum,
The network flips. Those exact same astrocytes ignore CCN3 completely. Instead, their Notch1 receptors connect with a totally different ligand called DLK1.
it's the same exact smartphone, but it gets a completely different software update based on its zip code.
Exactly. It ensures the astrocyte provides the correct metabolic support for that specific region.
Okay, final recap for you guys. The brain uses ligand receptor pairs like a local switchboard. Opioid pathways use lateral inhibition to quiet noisy neighbors, and receptors like Notch1 swap ligands based purely on location. So ask yourself, who are the ligands in your life that completely change how you receive and process information?
That is a very deep question to ponder.
Well, it's a deep dive. Okay, bringing it all together. If we can now map exactly how healthy cells alter their isoform trims and their communication networks based on their zip code, what happens when we look at a diseased brain?
This is the frontier. Historically, we've looked for broken genes or massive cell death in diseases like Alzheimer's.
Like looking for a demolished building.
Right. But this data suggests neurodegeneration might be much more subtle. The genes might be totally fine. But what if inflammation subtly shifts the neighborhood?
Oh, wow. So an astrocyte's Notch1 phone connects to the CCN3 network when it should be on DLK1.
Exactly. Or a neuron splices an automatic transmission isoform when the circuit desperately needs a manual transmission.
It's not a broken gene. It's just a zoning error. The right cell reads the wrong instruction manual for that specific street corner, and it triggers this whole toxic cascade of miscommunication.
Precisely. And thanks to this new RT&T-AMP-MERFISH technology, we finally have the camera resolution to see those subtle zoning errors in real time.
We aren't just guessing at the conversations anymore based on lights in the windows. We're actually listening in.
It is truly a new era of biology.
It really is. Well, thank you so much for taking this deep dive with us into the microscopic universe of the brain. Keep exploring, keep questioning the hidden layers of complexity in the world around you, and we'll catch you on the next deep dive.